massively appreciative, I know, from the families. So I'm going to hand over now to Jean-Christophe Roux. We have his presentation. So over to you. Thank you.
Thank you very much for this kind invitation. And I will speak a little bit about gene therapy and therapy in Rett syndrome. Probably it's a little bit, sometimes a little bit complicated. I try to simplify the informations, but if you have any question, feel free to ask me later. It's okay. And you can pass, yes, the next slide. Yeah, and just you can pass many. Sorry, yeah, just to the end. Just one more. Yeah, this is just to show the key dates in MSCP2 related research. In 1966, Andreas Rett identified first the Rett syndrome, and the Bengt Agbert in Sweden later on really described what is going on in Rett patients, because Andreas Rett only got 17 girls. I mean, it was fine, but Bengt Agbert really defined what is Rett syndrome later on. And in reality, in 1992, MSCP2 has been found by Adrian Bird. This is really one of my best scientists in the field, but he found it, but he didn't know that it was any connection between MSCP2 and Rett patients. And it was in 1999 that Uda Zogby made a connection between mutation in MSCP2 and Rett syndrome. And then after one point, I mean, very exciting, very interesting in the field, Adrian Bird and Rudolf Janisch, they created the first mouse model of Rett syndrome. And I mean, for many points, I mean, of course, mouse are limited, but we know that mouse models are very helpful to evaluate some problems, but also to try to identify some treatments. And after 2 more mutations has been found, CDKL5, FOXG1, but it's more for atypical Rett syndrome. Next slides, please. Sorry. Yeah, just to show you that the field is very dynamic. I stopped in 2014, but just to show you that it's a huge expansion. The number of MSCP2 publication is going up and up, and now we are close to 500 per year. It's huge for a rare disease. Just to show you that even if it's a rare disease, there are so many people, scientists, interested by this field, interested in this field. Next slide. Yeah, just you can pass here because there are several here. Just to show you, yeah, there are several projects in therapies. The first part of the first step is pharmacology, and people are more working on 1 or 2 symptoms, and they try to treat 1 or 2 symptoms like breathing, like epilepsy, locomotion, sleeping, and this is not, of course, the goal. No, it's not the goal is not to treat the pathology, but to treat at least 1 or 2 symptoms. And I will speak about another part. This is more, I mean, it's more phantasmagoric. I mean, it's more like fiction, but in reality, it's not totally a fiction. And the people are, many scientists are working to treat the cause of MSCP2. And this is, yeah, the goal is to treat the pathology. And I will speak about stop codon read-through, X chromosome reactivation, and replacement therapy, it's a gene therapy. We can go, yeah. It's better. Yeah, perfect. If you can put all again, again, perfect. Then for pharmacology, there are different targets that people are working on. Many laboratories are working on BDNF. BDNF, it's a sort of super vitamin for neurons. And the idea is we know that in red girls, they have the neurons are present. There is no they are not dying. Everything is fine, except that they are less connected and less. They are less exchanging chemical informations and electrical informations. And BDNF, this is a sort of super vitamin that is able to connect neurons together. And people are working in this way. And there are several clinical trials ongoing with cysteamine fingolimod. This is something that people are working on. IGF-1, this is close to insulin, the same family, but IGF-1 is doing more or less the same. I mean, it's not exactly the same, but it's pretty close to BDNF. And there are people working on IGF-1 because the problem of BDNF is not able to reach the brain easily, and this is a big problem. But IGF-1 is, if you inject to someone IGF-1 is able to cross the brain barrier and to reach the brain and to affect the brain and to increase the connectivity. And there are also some people working on neurotransmitters information. This is what we did before with desipramine, and we, in France, for example, we promote a phase 2 clinical trial with this molecule to improve breathing, to try to improve breathing dysfunctions in patients. And finally, there are some study involved for dietary supplements, curcuma, oils, and but there are many pharmacological trials, and probably currently there are 30 to 40 clinical trials ongoing or almost finished. So next slide. Yeah, now I will speak about something. I mean, the goal, I mean, it's exactly what parents would like to get. It's to treat the cause of the mutation, and I will present 3 types of projects. Next slide. Yeah, if you, as a parent, if you get the genetic diagnostic, sometimes if you see, next slide, just to show there is, the square, right square, you can see if you get this kind of genetic diagnostic with X, you see X arginine 168X or arginine 294X. It means X means stop mutation. Next slide. Yeah, and normally when there is no mutation, to produce MSCP2, a normal MSCP2 protein, you need to have a beginning. This is when you see the ribosome. I will not go deep in biology, but ribosome are reading a sort of book on RNA, mRNA. Okay, and a ribosome are going from left to right, and they are running from the ribosome to the normal stop codon. And if everything is fine in normal condition, you produce a normal protein with a full length protein. Next slide. But when you have an X mutation, it means that there is a premature stop. The stop is not at the end, but in the middle of the mRNA, and then when the ribosome is going from the left to the right, it will stop not at the red stop, but the yellow stop, and the protein will be small and truncated. It means that MCP2 will not be normal, but a little bit smaller and not able to do the normal function. Next slide. Yeah, next. Yeah, sorry, but now, yeah, perfect. And there are some molecules that we know that this is more or less antibiotics, and they are able to help the ribosome instead of to stop at the yellow stop, and they can pass through this yellow stop to reach the red stop, the normal stop, and stop exactly when it's necessary, and to create a full-length protein. This is very exciting because 40% of red girls, they have this kind of mutation. And this is something that is really exciting, but there are some problems now that people are, some companies are facing now. This is that this PTC or gentamicin, or there are several molecules that are able to do it, but they can make a sort of toxicity, cellular toxicity, and the benefit risk. I mean, it's right now it's not perfect, but there are many companies working in this direction, and this probably could be interesting. Not only, and you have just to understand that people are working in this kind of protocol because it's interesting for Rett syndrome, but not only for Rett syndrome and for company. Money is important, and there are so many companies interested by this kind of technology. Next slide. I will speak about chromosome X reactivation. I don't know if you are aware, but in girls you have two X chromosomes. One has a mutation, MCP2 mutation, and one is normal. And in patients, this is what you can see down in the picture. Half of cells, they have mutation, and they have X chromosome with a mutation, and one is a normal. And then, next slide. Yes, and there is something in biology, only one X chromosome is working. One X chromosome is compressed, totally compressed, and there is no possibility for this one to work. And this is why in red girls, they have half of cells totally normal and half of cells totally sick. And the idea is to find a way to reactivate the second X chromosome, because in red girls, this is sort of mosaic of cells, half of normal, half of six. Next slide. And the idea, next, yeah, again. And the idea is to reactivate the second X chromosome. There are some molecules able to reactivate the second X chromosome. And if you do it in sick cells, in the diseased cells, I mean, probably you can recover. But if you do it in a wild type cells, in the normal cells, nobody knows what is going on. But there are at least 4 big laboratories in the world interested to understand what is going on if we can reactivate the second X chromosome. But you have to understand that in biology, if only one X chromosome is working, I mean, this has been selected for many years, and nobody knows what's happened if the 2 chromosomes are working together. This is one option. Next slide. I will speak now about gene therapy. Gene therapy is an experimental technique that use gene to treat or prevent disease. In the future, this technique may allow doctors to treat a disorder by inserting a gene into a patient's cells instead of using drug or surgery. And but you have to know that the technique is a little bit risky because this is one shot. You inject a virus, and then the virus provide a gene, so a gene to treat the pathology. But later on, we have no option to date currently to correct if something is wrong. And this is still under study to make sure that it will be safe and effective. But the idea is it's very efficient. For example, if you have eyes problems or hear problems, because it's very located. The location is very tight. It's a special place. But when you have to target the brain, it starts to be a little bit complicated. Next slide. This you can pass a little bit because thanks. Yeah, you can pass. Thank you. Perfect. Yeah, there are two different types of gene therapy, but the first one I will not speak so much because there are not so many people working on it. It's the use of non-viral vectors, and it's lipids, liposomes, and people are working. Develop this technology. The idea, the good point is the quantity of DNA that you can put inside liposome. It's huge, but in reality, you can have toxicity and the capacity to infect the brain, which is the goal for Rett patients. It's complicated. But for viral vectors, people are working in two different kinds of virus. Adeno-associated virus, lentivirus, it works. It's virus plus DNA. It's very efficient. Quite efficient. You will see it's not perfect, but it works to infect the brain. But you can get possible immune reaction. It means that you can inject once. Twice it's possible. If you try third time, it's complicated because the body will try to fight against the virus, because virus, it's a virus, and for the body it's a stranger, and they fight. Next slide. But just to show you that in gene therapy, there are many clinical trials, but mainly in cancer research. And what we are interested, this is monogenic disease, because we are mainly speaking about MSCP2. And this is what is in blue, only 10% of clinical trials. But this is, the field is growing every year. Next slide. And just something that was very exciting, it was in 2007, because one question that we have to face, at what time should we start gene therapy? Is it too late or is it possible to start even late? And Adrian Bird, this is a fantastic guy working in Rett syndrome, and he created, this is not therapeutic approaches, but he created a mouse with, you can see down, this red, this is green square, this is a sort of cassette, they call, and you put a cassette inside MSCP2. MSCP2 is not working, and next slide. Only one, yeah. And the mouse is sick, and this is typical what you can see in Rett mouse model of Rett syndrome. The mouse is not breathing correctly, not moving. The mouse is really sick. And to save time, the next slide. Yeah, this is exactly the same mouse. After when you provide a molecule, the green square go down, disappear, and the mouse is totally fine, and it means that at least of course, it's difficult to compare to patients, but at least in a mouse model, it's possible to have a totally rescue. The pathology is totally reversible, which is good. It means that there is no death, no, the neurons are not dying, and it's possible probably to reverse part of the pathology. Next slide. The problem with gene therapy, this is where we have to reach the brain. Next slide. And obviously, it's easy if you inject the virus directly in the brain to provide a normal MSCP2 instead of one is not working properly. But this is probably will not be probably used because only in rare disease, when the patients are dying during the next few months or few years after, it's possible to use it, but probably it will not be possible, except if we are able to provide something very fantastic. We are able to totally rescue models. But today, I mean, at this time, it's impossible to think about this type of direct injection in the brain. Next slide. Again, next, yeah. The problem in the brain, I mean, we are using for gene therapy virus, and virus, the problem in the brain, we have the blood-brain barrier. This is a barrier that helps the brain to protect the brain from virus. And at the same time, we are using virus to correct genetic disease. And the problem we have to face that the blood-brain barrier is a block, diminish or reduce the capacity of infection. Next slide. And another problem. This is why it's a little bit complicated in Rett syndrome. We know that if you don't have enough MSCP2, 0%, you can have, if it's girls, you can have red girls, or sometimes for male, it's encephalopathic patients. But if you have 2 copies of MSCP2, it's not good. It means that the dosage of MSCP2 has to be really fine. If it's too much or too low, it's not good. And this is another problem. And next, and the next problem is to select. Yes, exactly, perfect, fantastic. And the next problem is if you have, like I told you before, you have patients with half of cells that are sick. This is the pink one. And half of cells are totally normal. And if I inject a gene therapy, a virus in the body, I will correct the green, the pink. They will be normal, more or less, depending on the dosage. But for the blue one, I will make a double copy of MSCP2. And this also, it's complicated. This is, I mean, there are some options. I would not go deep in this field, but it's a little bit tricky. Okay, next slide. Yeah, well, this is just to show that yeah, the virus is going. I mean, we have some virus that they have been created to identify neurons, or depending on the cells, some viruses are more able to infect one type, muscle, kidney, neurons, or glial cells. But yeah, now we have plenty of virus, and they go inside. And when they arrive close to what we call the nucleus, this is a pink part of the sort of ball, pink, and the virus inject inside the the nucleus MSCP2 just to correct, to challenge and to correct the genetic anomaly. Next. Yeah, next again. Yeah. Several people, I mean, several laboratories are currently working. I think there are more than 4 or 5 laboratories interested by gene therapy in Rett patients. And it has been found that, of course, if you inject directly in the brain, and if you inject at birth, before the pathology starts, it's very easy to rescue the pathology. This is good. But it means that we don't know yet how it could be translated to patients. Next. But if you do the same, use exactly the same virus for adult male mice or female male mice, it's a little bit more complicated. Yeah, it works, but so of course, more you start the genetic therapy. If you start very early, it's fine. If you start later, it's a little bit more complicated. Next. And yeah, thanks. And another story, another study has been done in another lab, and it's more or less the same. But I mean, the results are a little bit different, but it shows that yeah, gene therapy is working, it's fine, but we need to have some to improve the level of infection. And next slide. Just to show you what we are doing right now in our laboratory, we are injecting mice when they start to be sick, close. I mean, to have something close to the pathology. We don't want to treat the pathology, the mice before they start to be sick, but we want to wait till they are sick. And the little dot that you can see down, this is the number of infected cells that we got from the virus. Next slide. Next. Yeah, but the problem that we had to face, this is here. If you see the picture on the top, the little dot that you can see, this is a number of cells that are expressing MSCP2 in normal condition, and left at the right part, sorry, this is the number of infected cells that people, us, but other laboratory, found after infection with MSCP2 in a rat mouse model. You can see easily that the number of infected cells is too low to totally correct the pathology. And the second problem, if you inject too high dose of virus, we got some liver toxicity, which is not so good, obviously. And there is a need to develop technology to target the brain more and to avoid liver toxicity. Next slide. And this is why we start to develop, for example, in our laboratory, but a new technology with physicists to open the blood-brain barrier, the barrier that protects the brain from virus, to help the virus to infect the brain. This is what you can see down, the picture that you can see down. This is what you see in black. This is infection with a virus, and the left, this is without virus. And this is what we are trying to do to optimize, to improve the level of infection, to help to get something much better than before. Next slide. And there is another solution, and then there is a group, the same laboratory that created this mice that that was totally rescued, Adrian Bird. They were thinking, and the problem with virus, this is we are quite limited because the quantity of DNA that we can put bring inside the virus is quite small, and we have to. I mean, it's not perfect, to be honest, and this laboratory decided to work with a mini gene, a truncated form of MCP2, a small form of MCP2 gene. And what they published a paper showing that it was fantastic, because if you have even a small part of MCP2, it means if you have a small part of MCP2, you can have many other information around the gene that are really important for the gene to work properly. And this is something that we were not able when you bring the whole gene. But if you have a mini gene, you can have more information around before the gene and after the gene. And this is very important. And this is why they have shown with a mini gene, they found something. It was like a miracle, because the mice were living almost normal, no side effects, and everything were fine. Next slide. And this is why a company, Avexis, has been created to develop gene therapy for Rett patients. Novartis decided to buy the Avexis company, and they decide to go for a clinical trial with Rett patients, and not only Rett patients, but also another pathology. The name is SMA, but the clinical trials for Rett patients was planned for 2019, after 2020, the COVID came back, and then 2021, and next slide. But next. But the problem, the 2 weeks ago, it was a publication from Novartis, showing it was a shock from people working in gene therapy, because they published information, a commercial to explain that, however, with all preclinical testing now complete, the company decided that the data did not support moving into clinical testing in human participants. The specific data that led to this decision were not disclosed, and that was really a shock, because the clinical trials was planned, and we don't know exactly. We are trying to get information to understand what was going on, but yeah, we are waiting for more information. But we know that another company, Taisha, is still working on gene therapy and Rett syndrome, and yeah, we just have to wait to know first why Novartis stopped, and Taisha, what they plan to do to have something a little bit more, a little bit different from Novartis protocols. And next slide. Yeah. And finally, I just have to speak about this CRISPR-Cas9. I mean, this is the brandly new technology. 2 Nobel Prizes has been done the last year, one with a French girl and one with an American girl. And this technology, the idea is to correct a mutation. Instead of to provide a new gene, a gene working instead of one is not working properly. The idea is to provide sort of cutting, to cut before and after the mutation, and to correct the mutation, and this is fine. I mean, it's working really fine in cultures, in cell cultures, but in reality, to date, I mean, probably it will work later on, but currently it's complicated to infect, because the way to cut needs one virus, and the system to correct needs another one virus, and you have to have 2 co-infection together to correct the pathology. This is why to date it's complicated. If you cut, you don't correct, it's not good. And if you provide the corrections, the small RNA to correct without cutting, it's not good too. This is why it's a little bit complicated, but people are working in this way, and probably it will be an option in the future. And there are two big companies like Roux companies, French company, that are this company is very interested by this technology, at least in Rett syndrome. Next slide. Just for gene therapy, perfect. To demonstrate, I mean, the goal of gene therapy, this is to demonstrate that gene therapy can cure Rett syndrome in preclinical animal models. We have to optimize the way to inject in the brain, in the blood, in the spinal cord, to demonstrate that the treatment is safe and well tolerated, to minimize side effects. We know that they have this liver toxicity, immune reaction. This is off-target effect. And we have to monitoring the potential immune reaction or immuno, and possibly, which is pretty hard, to use immunosuppressive treatments. Next slide. This you saw before. The next. Just to show that the key dates, just perfect. We have, in the last 10 years, I would say, people have shown that, at least in mouse, it's possible to have a reversibility of Rett syndrome. The first clinical trial in patients has been done with IGF-1. In 2013, the proof of principle has been done with gene therapy. The first primate, I mean, it is in China. It's a little bit, I would say, hypocrite, because it's impossible to have a primate with a mutation, human mutation, in any kind of countries except in China. But this is an American groups, and with the Chinese scientists, she moved to China to create the model, and now even I mean, all the European and American groups, we are interested to understand what is going on in this primate. I mean, there is a sort of hypocrisy. I don't know if it's correct, but and in 2014, 8 active clinical trials. I think now we are close to 30 clinical trials only in pharmacology. And for gene therapy, I have no idea when really people will start, but this is one of the goals. X-ray activation, stop codon read-through, and gene therapy. This is where we are waiting for this kind of therapy. And next slide. Next, and just thank you very much for your attention. And if you have any question, just the people that and we are working in the group, and we are really yes, the French Association of Rett Syndrome is supporting us, and the French Téléthon is also supporting us for gene therapy, and the Rett, the IRSF Rett syndrome.org. That's nice to have all this kind of support. Thank you very much.
Thank you very much, Jean-Christophe. That's always huge.
Thank you so much for Laura, because it was fantastic. The way you pass, it was fantastic. Thank you so much.
Thank you, and really interesting. I know it's something all families, all parents are anxious to know more about, and and when it's going to be available and how effective it's going to be, and everything. I know we all follow it with a huge interest. Do we have some specific questions for Jean-Christophe? You see any hands up, Laura?
Feel free. I mean, I know that the notion or the biological points are a little bit sometimes complicated. X inactivation, double dosage, AAV, and feel free. I mean, there is no stupid questions, and you can ask any type of questions. And another thing, if you want to ask even by email, it's fine for me to answer. There is no problem, but feel free. There is no stupid question.
Okay, Mirlota, yeah.
Well, thank you, Jean, for your work. My question would be for this application of gene therapy into the brain about the intrathecal application. For example, what are the experiences until now? Because for some diseases it has been done already, and how dangerous it is actually.
Yeah.
Why did you say that it will probably not happen in Rett syndrome?
I would say, I mean, I think, to be honest, this is one of the most complicated pathology to treat. To really, to be honest, because first we have to target the brain mainly. First, which is complicated. Second, the dosage has to be correct, because we know that too much is not good and too low it's not good. Then we have to find something in between. And the third point is to find, since red girls, half of cells are normal and half are mutated, we have to find the way to target mainly the mutated cells, then and not the normal one, to not overdosage. And all these points are complicated, but there are some options. I mean, it's complicated to explain exactly, but there are some options. But it's complicated. But in reality, one good point also, this is that the dosage, I mean, too much is not good, 0 is not good, but we start to know that, for example, instead of 0, 0.3, 0.4 could be okay. And this is why people are working now with, instead of to bring back the full copy of MSCP2 to have 100% more, I mean, to correct totally. People are working with we call hypomorph. It's a low dose of the AV is providing a very low dose of MSCP2, but the idea is if you provide a low dose to nothing, it's better than nothing. You can improve, and if you provide a low dose to even normal cells, 1.1 to 1.3 will not be dramatic, and this is why people are working in this way. And also there is a company, Taisha, in USA, and they find a way, because one problem was for many projects of gene therapy, this is the liver. The liver try to protect the body from virus. Also, this is a way to block the virus circulating in the blood. And with MSCP2, if you provide too much MSCP2, the liver is sick. But there are some projects to decrease the liver toxicity, and we are working in this way to help to target the brain, to decrease the level of overexpression, and to decrease the liver toxicity. And science is going very fast, and I would say 5 years ago, I was not really excited by gene therapy, and now I think probably we'll find the way. But the problem is time. Well, it's time for parents. This is I know that you want to treat very fast, and the question is time, and it takes time to be sure, because they are so to be sure that it's safe and efficient.
Thank you. But my question was about this delivery directly into the brain. There are methods. There is this intrathecal methodology, and how dangerous it is actually, because it has been done until now. There is this surgery-like. There are special hospitals where surgeons are trained to deliver it. This has been done previously for other rare diseases.
Yeah.
So why? Is it also a potential way for Rett syndrome to avoid intravenous?
Yeah. No, this is an option. Definitively, there are the two main laboratories in USA. They are working this way with intrathecal. It means you inject close to the spinal cord and you inject in the spinal cord, in the cerebrospinal fluids, and to provide the virus instead of the blood. Then you avoid a little bit, because it has been shown, at least in one laboratory, that even if you inject in intrathecal injection of AAV, you can have a liver toxicity, a little bit less, but you can have. The problem is the spinal cord, the spinal fluids is not moving so much, and then the level of infection will you can reach. I mean, for people that are specialists of brain, but you can reach the brainstem, this part of the brain. You can go to the cerebellum, but it's really hard to reach the upper part of the brain. Yeah, which is complicated because in Rett we have. For a dream we want to target the whole brain, but in reality, by intrathecal injection, more or less you infect more or less the brainstem, the spinal cord, a little bit the cerebellum, but no more. I mean, it's at least it's good, but it's complicated.
It's not perfect either. Okay.
Exactly.
Thank you very much.
But
okay, thank you. I'm conscious of the time, so if we're okay, we'll take one more question we have from Mia, and then we will probably go into a break, but we put it to you, perhaps we shorten our break. But Mia, would you like to put your question to Jean-Christophe, please?
Yeah, I was a very nice presentation, thank you. And I was thinking, did you consider doing clinical trials on males with Rett syndrome? Because then you don't have the complexity of overactivating other X chromosome because they only have one.
Yeah, this is a very nice question. The only problem that we have to face that, in France, we have several patients per year with a male mutation on MECP2, but it's so fast, so quick that the pathology appear. They are almost dying when we start to get the genetic phenotype. I mean, technically, this is the best way to treat. I mean, that would be the best to start a project with a patient, with Rett patient, with gene therapy, just because we know that they will die, and this is an option to try to do something. But in reality, in 99% of cases, this is really too late when we start to know that there is a mutation somewhere, and it's the boys are almost dying, and it's really hard to. I mean, that would be fine, but it's really complicated to. Yeah. I understand exactly the point, but technically it's complicated. Yeah, we should be faster than we are to get the genetic the diagnosis. Yeah, good question.
Thank you. That's very interesting. Thank you. Perhaps Jean-Christophe, if you wouldn't mind putting your email address in the chat box, and then if people have got other questions, they can email you. So I'm just conscious we need to claw back a little bit of time. Thank you very much, both to Mia and to Jean-Christophe, for their presentations. Always good to hear from you.